welcome to the fourth lecture in our series, Innovation Gainesville. Our speaker today is Dr. Pat Lawman, who is the co-founder and CEO of Morphogenesis, Inc., located in Tampa, Florida, whose primary efforts are directed toward developing cancer immunotherapies using cellular and gene therapy technologies to fight a variety of cancers, as well as engaging the body's innate intelligence to fight chronic disease. Prior to founding Morphogenesis, Dr. Lawman was the Division Director of Cancer Molecular Biology at the Walt Disney Memorial Cancer Institute in Orlando, where she initiated and coordinated molecular biology, gene targeting, and universal stem cell projects. She's also held adjunct faculty positions at the University of Central Florida and the University of South Florida, and has served on numerous committees at the local, state, and federal levels. Under her direction, Morphogenesis has raised over $40 million through private equity and federal grants, and is currently in phase 2 human clinical trials for its cancer immunotherapy. Dr. Lawman also holds over 60 patents. She received her Ph.D. in molecular biology and immunology from the University of Florida's College of Medicine, where she also completed a postdoctoral fellowship in pediatric hematology and oncology. And as a fond remembrance, during her time at UF, while working on her degree in the lab I was working in at the time, she also managed to strong-arm me into learning how to scuba dive with her. So I thank her for helping me get to know our beautiful undersea world. So with that, I'd like to welcome Dr. Lawman, who will be talking to us today about her experience growing a biotech company from incubator startup to successful commercial venture, and a little bit about the activities Morphogenesis is currently engaged in. So welcome, Pat.
Well, thank you, Paula, and thank you for everyone joining today. This presentation is a bit out of the ordinary for me, but please bear with me. I'm not used to walking down memory lane, but hopefully it will be informative and just give you a little bit of personal history on how this all came about. So I'll try to share my slides here. And okay. Can you see that? Can everyone see that?
Yeah, all good, Pat.
All right, so this really is a journey into Neverland. I titled it Never Say Never. This is the best lesson I never learned, and it all has to do with survival in the biotech industry. So that's kind of the theme of our presentation today. So to me, I mean, never is a very emphatic word. It's final, it's decisive. Declares, this based on well-considered and logical decision-making, as it should be, right? Well, this is the origins of my never experience. In 1970, I went from Chipley, Florida to Gainesville, cultural and every other kind of shock. That was when 2-Bits was still young and energetic. The Hare Krishnas were giving free lunches in the Plaza of America, and that was my first exposure to halter tops and bell bottoms. So that was quite the experience. But I managed to get an associate degree, a bachelor's degree in anthropology, actually, and have one child, get married and have one child during that stint. But it kind of was a traumatic experience. So my first never was, okay, I'll never go back to school again. I'm done, that's it. So when a person says never, I mean, it means not ever, right? And so then, yeah, that was when I was young and naive. And I ended up back at the University of Florida. I did a degree in master's degree in plant pathology, and then I switched over to College of Medicine for my PhD. And in the meantime, I met Paula, as she just said, at the University College of Dentistry, where my major professor was her boss at the time, Dr. Arnie Blauweis. So he became my mentor, but my degree actually came from the College of Medicine at the then Department of Immunology and Medical Microbiology. But while I was in Gainesville on that occasion, one other thing happened, a pivotal event. Faculty member that was coming down from Saskatoon, the University of Saskatchewan and Veterinary Infectious Disease Organization was being hired into the immunology med micro. My friend Robin and I had a weekend away from graduate school in Key West. We were the only two people on the plane. Her seat was in the back of the plane. Mine was toward the front of the plane. But when we stopped in Miami and people were onboarding, the plane was quickly filling up, and this tired-looking gentleman came up to us and said, "Well, you're in my seat." And we said, "Oh, please, 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 kind sir, can you sit back in what was Robin's seat in the back of the plane?" Little did we know that was Michael, my future husband, and that's where we met, on a plane. So roundabout way of finding each other. He was actually born in northern Rhodesia, me in Chipley, Florida. But basically we came together, and we've been together ever since. We actually celebrate our anniversary, is that this week? Next week, the 14th, 32 years. But that was key, because everything that came then after that event. So number 2, moved to Gainesville, 2 more University of Florida degrees, a postdoctoral stint in hematology oncology, as Paula mentioned. And then I said, I'll never move back to Gainesville again. That's it, I'm done. I'm never going back. And so, yeah, we'll see how that worked out. But yep, you guessed it. After moving from the University of Florida to the Walt Disney Memorial Cancer Institute, where everything is about Mickey, even the monoclonal antibody clones that you see down here in the bottom right. Everything is about Mickey. We conceived the idea of morphogenesis, of a biotech company, a cell and gene therapy company that was going to change the world. And then we moved back to Gainesville, of course, to the Sid Martin Biotechnology Incubator. So that was number 3. It's like, I'm never going to say never again. But somehow these things happen. Maybe it has to do with Disney, Neverland, I don't know what. But anyway, Morphogenesis was born. If I had sound, I would like draw that out. So based on cell and gene therapy, we had grandiose ideas of enabling all cell and gene therapies. We had discovered at the University of Florida stem cell proliferation factor, which is a growth factor that allowed stem cells to proliferate without differentiation. From that, we were able to grow very primitive cells, hematopoietic epidermal stem cells. And we created monoclonal antibodies to very early stem cells. That allowed us to make a cell separation device to separate out these stem cells. And then we had a way to actually create universal stem cell banks that would not be rejected when used as therapy. And then we sort of went the other way of manipulating these cells to develop our immune effects cancer immunotherapy. That's where we have focused most of our efforts in the last few years. But moving to the Sid Martin Biotechnology Institute was where Morphogenesis was born. It only took me a year, and I wrote this article. It was a women's science group that invited me to write this, and it was very reminiscent of giving birth, having at this point given birth to 3 children. But I just call out the conception is that, you know, that was the fun part. The first, second, and third trimesters not so great, and then birth in itself. And I call out this little bit here is because fact is that we laid everything on the line when we started this company. By this time, the 6 or 7 other people that had gone with us to start Morphogenesis in the incubator from the Walt Disney Memorial Cancer Institute had found other jobs. It was basically Mike and I were left there for the last year or so. And really, I mean, this was an all-in effort. And we're not people who like to gamble. I've been into a casino once in my life. I came out with the same $5 bill that I went in with. So I'm not the type to gamble, but this was the biggest gamble ever. So Morphogenesis was born. We moved to the incubator, as I mentioned, and we stayed there for 3 years. We had two laboratories. We were very productive. Actually, while we were there, we filed 3 patent applications, which were issued. We submitted 2 small business innovation research grants, one to the National Cancer Institute and one to the National Heart, Lung, and Blood Institute. Both of those were funded. We completed the work on those, phase 1, but we had to leave before we could file for the phase 2. So that was sort of like a stunted growth. We were within weeks of being acquired by other biotech companies twice. Neither time worked out. But then after 3 years, being one of the first tenants at the Sid Martin Biotech Incubator, actually, at that time, people thought that you should have done your IPO, graduated, and just be off and running. But we graduated only to become virtual. So we were a virtual biotech company for a number of years until we ran out of food, and Mike took a job at the St. Joseph Children's Hospital here in Tampa. That was so we could eat and have a place to live. So that's what happened there. Like I said, the incubator was just starting when we looked out our window in our office. This is the picture that we saw. There were cows there. I don't know how many of you remember that when it was like that, but I don't think the cows are there anymore. So number 4. This immunotherapy thing, how could it ever work? It was kind of way too early to even think about how this would work, but we had an idea that we wanted to draw the immune response to tumor cells and expose them as being nefarious, just like a bacterium or a virus would be. So maybe this time saying never was what guaranteed would happen. So the challenge with immunotherapy is to balance. It's a balance in trying to create a positive immune response, but not an overblown immune response. So those of you with a science background, you'll know that when you have a bacterial infection or a viral infection, there is an acute inflammatory response that is generated. And this is a positive thing. It kills the pathogen. So this is a protective thing. It's very controlled and is normally localized to whatever the insult is. But sometimes our body goes under stress. We're constantly exposed to these agents. And what was an acute inflammatory response that was protective then becomes a chronic response. And this is one way that cancer is actually established. So it's initiated and established. And so the tumor microenvironment for solid tumors becomes a chronic inflammation kind of thing. And this is a pathological response. It's uncontrolled, and it can go systemic. And what we do with immunological intervention is try to balance this. So some of the therapies that are being developed now have hyperactivated T cells. So like CAR T, these are T cells whose T cell receptor is engineered to bind to a single epitope of a particular antigen. Most of these CAR Ts have been developed, as you know, for liquid tumors, so leukemias and lymphomas, because those are practically clonal diseases. And so if you choose a single antigen, most of a B-cell lymphoma will have a CD19 or a CD20 molecule associated to it. So you have this vast number, millions and millions of T cells that attack this one epitope on tumor cells, and it kills the tumor cells. It's very good at it, but it also creates this severe inflammation, so cytokine storms and other things that go along with it. So it actually can then become a pathological, uncontrolled, and systemic response. And so what we're trying to do is balance this. We want to create an immune response, but we don't want it overactivated either. So if you have activated T cells against multiple tumor antigens, you can create this acute inflammation, inflammatory environment in the tumor, and then you kill the tumor. So this then becomes protective, it's controlled, and it's localized to the tumor. And when I say tumor, this could be distal tumors, but basically there's no bystander effect. There's no overblown cytokine response. So this is really what we're trying to do, creating this balance. And we've chosen a kind of odd way to do it, but I think it's a pretty simple way. We take a single bacterial gene, put it into a patient's tumor cells, and have it expressed. So the gene that we chose was actually discovered by another University of Florida faculty member, Dr. Michael Boyle, who generously allowed us to use this. So in the group A strep, this is basically what it looks like. It's a transmembrane protein. It's out on the surface of the strep. It's a dimer, alpha helical coil, but it has a lot of biological properties as well. So this looks to the immune system like a bacterial infection without it being a bacterial infection. So this is a single protein, as I mentioned, that's on the surface of the tumor cell. And why is it on the surface of the tumor cell? Over to the right, you'll see this is the predicted conformation in the lipid bilayer of a tumor cell. So serendipitously for us, the same signal sequence that allows this protein to become lodged, going from the inside of the strep to the outside of the strep, the signal sequence. Inside that bacterial sequence, there's a mammalian consensus sequence for signal transportation. Likewise, in the transmembrane portion of the bacterial sequence, there is also a mammalian sequence. So that's why we think this protein is actually expressed on the surface. And we think this is key. So you look at this Western blot analysis, it's in the cytoplasm, it's also in the membrane, this flow cytometric analysis. And then on this immunofluorescent picture of a lymphoma cell, you can see EMN55 is certainly on the surface. So what does that do? I mean, as a double-stranded DNA, which is the current formulation that we're using, a plasmid DNA with only this gene in it, it has some other things that will make the immune response respond. But basically, what we're looking at here is this eat me signal. So when this is actually injected into a person's tumor, and this is the formulation that we're using now is directly injected into the tumor. It expresses this protein on the surface, and I've put it in this diagram as a red flag. Mike hates the red flag, but it's really an easy way to picture this because the tumor cells are in stealth mode. They're there. We know the immune system can get rid of them if they can only see them. And so we just put out a beacon, a flag that attracts the immune system to it. So these professional antigen presenting cells, these APCs, come along and they see this bacterial protein and they think, yikes, this is a bacterial infection. We need to do something about it. And then as antigen presenting cells do, they eat the cells, they get the virally infected cells, bacterially infected cells, they eat them, and then they display the antigens, all the epitopes that they have digested from the tumor cells, proteins. They display them on their surface in the context of class 1 and class 2 MHC, major histocompatibility complex. And so there's, for every epitope that is displayed correctly and appropriately in this way, there is a T cell and there's a B cell that will recognize that. So that's what happens is that these T and B cells come around. If they do recognize what is known as their cognate antigen, that's the only epitope they can ever recognize, and they can only recognize it if it's presented appropriately, then they become activated. They start proliferating and they start trafficking, and then the T cells kill whatever cell bears that particular epitope. And so they can travel throughout the body. They'll kill distal tumors as well as they kill the injected tumor. So this is a systemic response. And so this series, we call it epitope spreading, continues. So we gave it our bacterial antigen that spread the immune response to the tumor cell antigens. And then as more antigens are released, those T cells become educated as well. This also happens to the B cells. So the B cells have receptors as well. So they become activated when they recognize their cognate antigen, and then they proliferate and start making antibodies. And so we know that B cells make IgM to begin with. That's the first antibody that B cells make. So that is an indicator of a primary immune response. And then as the immune response matures, the B cells switch over then to IgG. So what we did, and I put this as real, this is real data here. These are experiments that we've done using patient-derived blood samples. So these were melanoma patients that were metastatic, very advanced. Most, all the patients had progressed on multiple types of diseases. They were all elderly, they had multiple comorbidities. But basically what we did was we took blood before our, what we call immune effects was injected into the tumor. And then at some time point after, usually about 4 weeks after, because that was the end of the study for these patients. And basically we put their plasma containing antibodies on a microarray. This microarray had over 4,000 epitopes from these known melanoma antigens. So if you see here, tyrosinase, MYC, MAGE1, all the way down to BEIJ1, those were arrayed in overlapping antigens. So there was no epitope within these melanoma antigens that was left off of this microarray. You can see over on the right the number of potential peptide sequences that were available. And so what you're seeing here in these bars, this is the differential expression. So these patients had no antibodies in their blood prior to our therapy to any of these epitopes. And now these, the different colors here are different patients, IgM and IgG. The number of peptides now that they have made antibody to. So this really just shows that when we inserted this one bacterial antigen, the response then spread to multiple tumor antigens. And these are only the antigens that we know about and we tested for. So there may be an untold number of other patient-specific epitopes that were there that could be targets for the immune response. And so basically that's what our therapy does. It's an off-the-shelf product. It's a plasmid DNA. Our next generation is a messenger RNA. But basically the mechanism of action is the same. It's an off-the-shelf product, goes into the tumor, and it creates this very personalized immune response and a broad, multivalent, systemic, and sustained response. And we have loads of data to corroborate what I just said, but I just wanted to show you that this is the mechanism of action. And we tested this in multiple species over the years. The first study that we did was a neuroblastoma study, very aggressive model. Mice die within a couple of weeks if they're not treated. As you can see from the survival curve, this therapy worked very well. This is a syngeneic induced preclinical murine model. We then tested it in dogs with canine lymphoma, again, a very aggressive disease. If dogs are not treated, they have just days to live. Historical controls of 7 days to 59 days, so eight weeks on the outside. And you can see that we had made a significant difference, either as a monotherapy or in combination with standard of care. We then did an equine melanoma study. This is a controlled USDA study where 22 horses were injected in 3 lesions, multiple time points. And at 35 weeks, you can see the tumor reduction in 2/3 of these animals. Then our colleagues at Moffitt did a murine melanoma model, basically all the same thing. We saw an additive effect with anti-PD-1. But basically through all of this, what we saw was there were no adverse events. There are no serious adverse events. And so we felt like going into humans that we had a lot of safety data. But my goodness gracious, will we never make it to human trials? That was the main thing. And I don't know. So when we first filed our IND, we thought, okay, we're on pins and needles. But we had so much animal data that the FDA really had no comments. I mean, that's really unusual when you file your first IND for it to come back with no comments. They said, go right ahead, first in human. So for good or for bad, we chose to do our proof of concept in humans in skin cancers. And living in Florida, we all know about skin cancers, but I just wanted to put things in perspective here. These are the most commonly diagnosed cancers in the US. So if you look down here, skin cancers, the number of newly diagnosed skin cancers in the US in 2019 was 5.5 million cases. If you put all of the non-skin cancers together, it's 1.7. So there's so many more people, so many more cases of skin cancers. But really, the efforts are up here in the top corner here. It's the regional metastatic diseases that people are concentrating on. So Merkel cell, for instance, I mean, there's 2,000 cases a year in the U.S. and with only 35% of those 700 cases that is regional metastatic. Melanoma, 13% regional melanoma. So there's only 11,000 patients of this. Cutaneous squamous cell, 5% regional metastatic. Basal cell, only 0.5%, but you can see the numbers go up here. I mean, there are 21,000 people with advanced basal cell, but there's really not much that people are doing for them. So we wanted to be able to show a proof of concept where all the competition is basically in regional metastatic melanoma cases. There's tremendous competition. All the big academic clinical sites have ongoing trials. I think Moffitt has like 12 melanoma trials going on. But nevertheless, we chose to do that. It makes really slow patient enrollment, but that's what we did. But we also chose this because this current formulation is a direct injectable. So we needed something with accessible tumors. We knew that people were going to look at how this worked in combination or compared to anti-PD-1s or PD-L1s, so checkpoint inhibitors. So these therapies have been approved for all of these skin cancers. So this is, like I said, for better or worse, this is where we chose to do our proof of concept. Our first human trial was in cutaneous unresectable stage 3-4 melanoma. This trial is completed. It was for 6 patients, very simple protocol. They came in, they were injected. 4 weeks later, we had a follow-up. And really the primary objectives was to look at the feasibility of intratumoral injection and look at the safety before we went any further. So again, this trial is now finished. It was a single site trial at Moffitt Cancer Center. Most of the information that we've gained though is from like I mentioned, the blood and the tissue from these patients. And so we did extensive correlative studies and trying to untangle what's going on in the tumor microenvironment, what's going on with the immune system. So we took tumor tissue before and after. So this is a differential response. We did transcriptomics, nanostring. We analyzed over 770 genes, again, for each patient before and after. We also did a multiplex IHC with these various biomarkers. For the blood, we did proteomics. We did an O-link panel. So this is actually looking at the actual proteins in the blood before and after. We did 2 panels, 92 proteins each. And then the antibody study that I just already mentioned to you. And so basically, this is what we saw in this melanoma study. Again, these are the feasibility, safety. So again, these are the patients here. They had long medical histories. They're all elderly. They had progressed on multiple therapies, like I said, BRAF, TREC, oncolytic viruses, Ipi, Nivo, Pembro, all of these checkpoint inhibitors. Patient number 1 was about to have their leg amputated for melanoma. This was first in humans, so this patient had 1 injection and 1 lesion. That patient is now 29 months out, doing well. Went on to respond to checkpoint inhibitors. Again, there were the injection site reactions, redness, pain, maybe slight bleeding. There were no product-related SAEs. There were no antibodies that indicated any kind of autoimmunity. And then they all went on to various therapies. So basically, this was our little contained study, but we have been able to follow these patients out. This last patient has not gone in any therapy since their injection. This is the only patient that had injections at 2 time points. But the results of the correlative studies, just in a high-level summary, was the molecular biomarkers, both at the gene or transcription level and the protein level, indicated across the board immune localization to the tumor. So chemotaxis, bringing the immune cells to the tumor microenvironment, antigen presentation, antigen recognition, T-cell priming, T-cell activation, and tumor cell killing. This happened in all 6 of the patients. So even though it's a small patient number, we're very excited about these results. The anti-tumor antibody response, I already basically told you that the initiation of the immune response, IgM, effector response, IgG, epitope spreading, same epitopes, totally different profile before our therapy and after the response to our therapy. So there was no way we could have actually predicted which one of these antigens that each patient was going to look at. And that was in 6 of the 6. Cellular markers, there was a decrease in SOX10, which is a melanoma marker, in 5 out of the 6 patients. There was a decreased cellularity in 4 out of 6, and there was a statistically significant decrease in the Treg CD8 ratio in all 6 patients. That's a prognostic factor. So we're very excited. We're very happy for that trial to be over. It took a lot longer than we thought it was going to. We learned a lot. We then now are positioned for phase 2, late stage melanoma. So what we learned was that when you create this broad immune response, if you then give a checkpoint inhibitor, you take the brakes off of those cells and allow the immune response to work. It does very well. So that's how we designed this phase 2 study. The study should be opening in the next couple of months. This is for PD-1 refractory patients. This will be at 15 clinical sites, and it will have 3 arms of the study. So whatever checkpoint inhibitor the patients are progressing on, one cohort will stay on that while taking our therapy. The other one will be taken off of that and then put back on after several doses of our therapy. And the third will be just a monotherapy to see how all these compare. So this will be an objective response rate according to RECIST. This will have all of the efficacy and safety endpoints. We just received recently orphan drug designation for this, our therapy, to treat stage 2B to 4 melanoma. So that was exciting. And then last Thursday, we received fast track designation for this patient population for refractory patients in melanoma. So both of those, we should be having a press release about that this week. In the meantime, we started another trial. This is for non-melanoma skin cancers, cutaneous squamous cell and Merkel cell carcinomas. This is a 20-patient trial. It's ongoing in 3 clinical sites. We're about to open the 4th clinical site now. This is a monotherapy. There are 3 cohorts. First cohort received an injection in up to 3 lesions at a single time point. Second cohort, first patient has been dosed. We have 2 squamous cell and 2 Merkel cell patients have been treated so far. And then the third cohort will receive injections at 3 time points. So this is really our dose escalation. I mean, we can only fit so much plasmid DNA directly injected into a tumor. So we do multiple tumors, 1, 2, or 3, and then up to 3 time points. So at the max, a patient could receive 9 injected lesions over time. So we think that the biggest opportunity, though, that I mentioned before is not in these regional metastatics. So over here on the right, you see that is only 1.6% of all the skin cancer patients. Most of the skin cancers are localized. They're very early stage. But as you can see, the products that have been approved by the FDA and are sold are for the regional metastatic. So they're for early stage Merkel, early stage melanoma, early stage squamous, early stage basal. There's nothing. And there's several reasons for that. These immune checkpoint inhibitors are very expensive, as you can see the annual cost here. They're also very toxic. And so who is going to treat an early stage skin cancer that can be surgically excised with that? So really there's not very much in the immunotherapy space here. So we hope to capitalize on that. And so hopefully next week we'll be starting this study. And this is a very simple study again. This is a neoadjuvant study where the standard of care for these patients is surgical excision. And so we'll just intervene here with a single injection. 4 weeks later, we'll take a biopsy and we'll look at pathological response as well as safety. So this is an efficacy trial, but it's not a registrable trial, registration trial. We just want to see if we can make a difference here without, upsetting anybody's apple cart. We're hoping that this trial will recruit a lot quicker. We're going to dermatological clinics here in Florida that see literally hundreds of these patients every week. And there's no competing trials like there are for melanoma. So hopefully, knock on wood, we'll recruit this and see statistical differences that then will inform a registrational trial. Our pipeline beyond the skin cancers, again, those are proof of concept. We plan to segue into head and neck, gastric, colorectal. We just received a fairly large grant to develop a certain formulation for cervical cancer. And then we've been working with the Karolinska Institute to do a targeted delivery for diseases like multiple myeloma that are not directly accessible for injection. So we feel, and I know we're prejudiced, but we have what could be a paradigm shift. Right now, cancer immunotherapy is based on cell therapies, the CAR-Ts, and also the checkpoint inhibitors. The checkpoint inhibitors take the brakes off, if you will, of already activated T cells. So there has to be an underlying immune response for a checkpoint inhibitor to work. That's what our product does. So as far as safety, this is an intralesional injection. It's not systemic. It doesn't go everywhere. It doesn't cause toxic side effects everywhere. We don't have to precondition a patient. We don't have to take out their blood, their T cells, their B cells. There's no possibility of any kind of systemic infection or a viral recombination. This is just a single gene in either a DNA vector or ultimately an RNA. And so this what makes this first line treatment very feasible for us. It's not trivial to produce plasmid DNA or an RNA, but it's a lot easier than doing a cell therapy or some kind of autologous thing where you have to take out the cells, do a genomic analysis, predict what they're going to respond to, synthesize a protein and put it back. We believe that it will prove itself to be effective like it was in the animals that have been treated. For one thing, it leverages the mutational load of a patient's tumor. So this was really the main problem for chemo and radiation therapies. If these these tumors are evolving, they're mutating. And so as soon as you irradiate one or you give them a chemical, survival selection is going to sort out the ones that can become resistant. And then those go on to expand. So our therapy, actually, the more targets there are, the more mutations there are, the more antigenic targets the immune system can see, the better. So that's what I mean by leveraging the mutational load. And as you can see, it induces a multivalent immune response. And it primes this response to literally hundreds. I didn't show you the numbers, except in that one little graph, but this is literally hundreds of newly recognized epitopes that these patients' immune system had not seen before and only receiving our therapy. And to me, a lot of companies and the hype around the oncology industry leave out this last thing, which to me is really important to patients. It's like it needs to be affordable, or you're only going to get, I mean, a handful of people that are going to get it. When you have a therapy that costs $500,000 and then it costs another $1 million to treat the side effects, that's for one patient. That leaves out most of the world. So it's really, really important to us to have our therapy become affordable so that you can treat people that would never have had the chance for any kind of cancer therapy. So again, this is cancer agnostic. I mean, it doesn't matter. It could be skin cancer, it could be gastric cancer, it could be pancreatic cancer for that matter. And we have developed in-house, we have our own manufacturing facility here. Paula, you probably didn't know that as well. But we're going to go full GMP here in a few months to produce all of our plasmid and whatever other products we develop in the future. But it's very cost effective. And we don't need any patient information. And so far, knock on wood, there's been no supportive care. This is a 30-second intralesional injection. The patient is watched for an hour, and then they go shopping or go play golf or work on a car engine like some of our patients have done. So that's the morphogenesis story to date. I appreciate your attention. I'm happy to answer any questions, and thank you for your time today.
Okay, well, Pat, thank you so much. That was really informative, and it's great to hear what's going on in morphogenesis. Do really appreciate you taking us through the aches and pains that were the growing part of that. I mean, it's you know, I think good for people to understand it's not all rainbows. You know, there is a little pain involved in growing, I think, a bit any business really, but especially, you know, in your sector. I think the ImmuneFX strategy just is so fascinating. So why haven't you been bought by Pfizer or any of the giants? I mean, they should be all over that.
Yeah, I ask myself that daily. We haven't really gone, when we did a few years back, think, oh my gosh, why isn't BMS, why isn't Merck all over this? Because checkpoint inhibitors only work on 30% of melanoma patients. And then a large part of those patients become refractory to those. And so that leaves 70% that never respond in a situation that's optimal for checkpoint inhibitors. Melanoma, highly immunogenic tumor. So their $7 billion in revenue from Keytruda, for instance, is only 30% of what it could be. So you'd think they would be, but you know, they have so many people. When people realize something, they jump on the bandwagon, and so it's like 1,000 combination trials in PD-1 now for mostly melanoma, fortunately. But so they, you know, I can't speak for the big pharma companies and just the couple of people that we talk to. They want to see patient data. They want to see, you know, show me the data. I mean, we've heard this all before. We hear it all the time. And unfortunately, some of the choices that they've made to combine with haven't panned out. So that just makes it harder for us to say, oh no, we're different. And they're going like, what? You're a little company in Tampa. We've never heard of you. What's this with a bacterial protein?
Right.
Yeah,
So,
it's not easy.
well, so I'm just curious, and I'm sorry if I missed this. So, how do you ensure since you do direct injections into the tumors, how is it that you ensure that it stays just directed to the tumor cells and not to any of the patient's normal?
Sure. That's a good question. I get that a lot. The truth is that you can't. I mean, you're going to inject it. It's a liquid. You hope that you formulated it so it's taken by the cells efficiently, but some of it's going to leak out. So we actually, before we started this, the FDA asked us to do a biodistribution study, which we did. So that was a very extensive study. It took us like 6 months to do. We looked at a time course injecting into tumors in mice. We looked at the uptake immediately and then over time, not only in the tumor, but all the major organs. So we had 11 different organs. So basically, some does leak out and some does go to other organs, but it's so, I mean, it's like we were at the femtomole for detection and maybe a few copies, but it's so short-lived. And then the other way to look at it is that if a normal cell did, I mean, because the FDA has approved us to do the intranodal injections. So there's a lot of other cells, immune cells in like a lymph node. But the FDA realizes that, well, people are transfecting immune cells to make a cancer therapy. And so they wanted them to produce an antigen. But if you were to just in a normal cell, like say you were getting a COVID vaccine, it goes right into your muscle and you think the RNA is going to have to, like you're going to get, right, Pfizer, or you got the Pfizer. So the mRNA is in your normal muscle cell. So it's, if we brought the immune system in, the antigen presenting cells eat the tumor cells or the normal cell and display the peptides. Those are normal peptides. They're normal antigens. And theoretically, in the developing thymus of a fetus when we were small, any T cell or B cell that was could recognize a normal epitope was destroyed. Clonal deletion. Is that 100% no? So that's why we monitor every patient for anti-nuclear antibodies, because the one big side effect that could occur from this is autoimmunity. I mean, as a strep biologist, you know that probably better than anybody.
So you haven't seen any issues with that?
Never. So over 3,000 doses had been administered to animals before this. And it's the #1 thing that we're looking for. I will say that checkpoint inhibitors have a bad reputation for autoimmune disease because they're taking the brakes off of whatever has already been, it's the good, the bad, and the ugly, the T cells that are just hanging out there that do recognize normal epitopes. So knock on wood, we won't see it. We haven't seen it, but we're on the lookout for it. We don't think that it's a major problem, especially for these patients that are terminal, that are on maintenance therapy. They, it's we'll see.
Yeah. Well, and so what? Let me just see if there's any other questions. So I'm not hogging the floor here. Okay, not yet. So I guess another question would be is ideally, how early do you want to start this process of when you want to start injecting patients? I mean, I can see where it could be pros and cons for late stage versus early stage, but ideally, once this is out in the marketplace, what would be the optimal time to start this process?
Well, to me, the earlier, the better. The immune system is intact. The tumor burden is at its very lowest. Because remember, when the immune system is involved, these T cells and the antibody interactions, however that works, is a cell at a time, a tumor cell at a time. So it takes time. And the more tumor cells there are, the longer and the more possibilities of escape. So the very earliest would be optimal. So I try to keep myself contained, but if we could prevent those late stage cases, why not? I mean, our body does this every day. We're in Florida, we go out in the sun. You know, UV radiation is responsible for most of these cellular skin cancers. There's a mutation, right? But it makes something so different that the immune system takes it out and it's gone when it's a single cell. We don't even know it. How many times in our life has that already happened? Right. It's just the ones that become really sneaky and can establish themselves and stay under the radar of the immune system that get by. So to me, the earlier, the better. But as I said, the immune system is more intact and more able to actually counteract this. But all along the way, even if you get to the patients who have failed or progressed on every other therapy, this can still work. I mean, these patients in our melanoma study, as I said, they were elderly. Some of them had 2 or 3 different kinds of cancer. They had progressed on everything that had been thrown at them. So but they still, I mean, we can't say, oh yeah, we cured them, but they're still alive when they most likely would not have been. Yeah.
Right. And so is there a minimum sort of threshold of tumor size that you need? So I would think that if you have too few cells there, or maybe not, but I was thinking if you have too few cells there, you really don't get enough of an immune response. But what do you have, like a minimum threshold there that you need to be effective?
It's more like a practicality. It's like how much liquid can you get into a certain size tumor? So the choice was inject as much as you can get in, regardless of the size tumor. But then the dose is different for every patient. So we decided to just stick with something that we could get into a 3 millimeter. That's like the minimum, a 3 millimeter in diameter tumor. So that actually holds 100 micrograms of plasma DNA and 200 microliters. So the injection is made in a fanning technique. So the needle is put in and then some is injected and then it's not taken all the way out, but it turns around and then through the whole tumor like that. So if you do it slow enough and correctly enough, it's absorbed. This particular formulation is in a cationic polymer excipient, so it sort of condenses the plasmid DNA, allows it to go through the tumor cell membrane, and then allows its release once it's in the endosome, and so it releases in the cytoplasm. So far that's worked out really well. As I mentioned, the dose escalation really is on the number of tumors that can be injected and over the time points. So we're working all of those things out because we really don't know. I will say that the one patient on the melanoma study that received doses at 2 time points, the antibody response was almost equal to the first injection, but it was to different epitopes. Very interesting.
Yeah. Okay. All right. Well, I don't see any additional questions, so I think I'm going to leave you with this final question. What would you have as advice for anybody thinking about starting their own biotech company? You had to go back in time and do this all over again.
Think long and hard, get lots of advice, talk to lots of people who have done it before, and make sure that it's something that you can stick with. Mm-hmm. It's rewarding, but sometimes when you say something like it's hard, that doesn't really describe what hard is. And I'm always reminded of when I was a PhD student, and with you, it's like clone a gene. That sounds so easy to clone a gene, and nowadays it's a lot more easy than it was back then, but there's a lot that goes into that. And it's to describe how difficult it is sometimes when you're trying to meet payroll and you go months and months without a check, trying to find investors that are interested, trying to create a story that will be interesting to them. getting through the regulatory hoops, while running a business, doing OSHA guidelines and having SOPs and all the things that go into it, accounting, dealing with the SEC. I mean, there's just so many aspects to a biotech company. It's probably one of the most complicated businesses there is to start and then to run, and it's very expensive.
And do you feel that your time at the Sid Martin, do you feel that was an advantage to getting started as a company? And I don't mean to put you on the spot there, but there was probably some pluses to having something like an incubator to kind of get a toehold on?
Absolutely. I think too, to nurture these ideas that come out of always out of academia, there needs to be a landing place. You need a safe place to start, something that doesn't cost you a lot of money. So you don't have an upfront expense of facilities and manpower and all that kind of stuff. That's really, really important. But it's also important later stages. Like when we started to think about, well, we want to do a human trial. Well, how are we going to have product? Do we have to make a fully GMP product? Because if you go to a CMO, a contract manufacturer, and ask them to give you enough DNA to treat 6 patients, I mean, by the time you get done, it's like several $1 million.
Yeah. Wow.
So who can afford that? And to buy all the equipment and make that investment, that's huge when you don't even know if it's going to work or not.
Right.
So to me, one of the missing pieces in this whole thing is like you have an idea, you take it through its preclinical, and then there's a gap between the preclinical and the clinical. If there were incubators that would say, okay, we have all this equipment, come in and make your stuff for what you need it, and then leave, to me, that would be like really a good way to nurture that transition between having preclinical data and then you're first in human. So if I had the money, I would do that.
Okay. Well, great advice. And Pat, thank you so much for speaking with us today. And if we don't have any more questions, thank you again. We appreciate your time. And
well, I appreciate it. I hope everybody got something from it, and thank you for allowing me to do it, Paul. It's great to see you.
Great seeing you, absolutely. And everyone, we'll be here again next week where we're going to be talking with Wanda Eugene, who's the director of the Collaboratory for Inclusive Entrepreneurship, titled Stuck from the Start. So she's going to be telling us about some of the roadblocks for women and minorities in developing in the biotech sector. So we'll see you all next week.